Okay, let's do this. Hello, good afternoon. It's Friday, yay! So day 18. Blimey. So we've been at this for a while, and I think we're doing quite well. We've done lots of basic introduction to Linux, and we've then downloaded some data from Uniprot and explored that data a little bit using multiple sequence alignment. And this week we've been downloading data from the sequence read archive. So we're downloading sequencing data, and then next week we're going to move on to processing that data. So I've got a couple of little bits of housekeeping first, and then I think I'm going to spend the majority of today talking about specifically about FastQC. So we had a brief look at a FastQC report yesterday. We're going to take a slightly more detailed look at one of those reports today and talk a little bit about the assumptions that FastQC makes and so forth. So first bit of housekeeping by way of a little bit of a lockdown diary entry. I wanted to share with you the misery that has been my afternoon. So I've had the last 2 weeks off work, as many of you know. So I've been full-time childcarer, and this morning via those lovely people at Amazon, a parcel arrived for my children containing some Easter games, and one of them is what can only be described as a flatulent pig. And the idea of the game, for want of a better word, is that you pass the pig around until it passes wind. And so I just wanted to share with you the misery that was my afternoon by playing you the flatulent pig. So with apologies in advance, here we go. There you go. Isn't that fun? All afternoon I've had that pig singing. Anyway, I just wanted to pass on that particular piece of misery. So to get on with today, what we're going to do, as I said before, is talk in a bit more detail about FastQC, about the reports that FastQC produces. In order to do that, let's do a couple of things first. Let's check that. Oh, there was one other piece of housekeeping. Sorry, I'm distracting myself. Right. So here's my VM. The other couple of bits I wanted to talk about actually briefly were whether we can resize the virtual disk for our virtual machine, because if you recall, when we made our VM, we only gave it a 10 gigabyte hard drive, and increasingly that's proving to be inadequate. We're slowly but surely running out of space, or certainly I am. So I've got 77% full use on that disk. So it's probably going to be the case that we're going to have to expand it at some point in the future. I have explored how to do this. It certainly is possible. And I think the process is slightly different depending on whether you're whether your host machine is a Mac or a Windows machine, but it is possible to expand that disk. So I think we may look at doing that in a future lesson, maybe next week. I don't necessarily have time to do it today, and I haven't tested it yet, and I don't really want to do it without testing it. So I'll look at doing that, I think, next week. I think that's probably quite important. The other thing was, I had talked yesterday about a more robust way of making sure that we've downloaded the data that we wanted to download. And so I set off doing that yesterday evening. So I just want to show you what I was doing week 4. So I made, sorry about the mess of my files, this is part of what kind of results from me doing this kind of experimentation. I'll find a link for the pig. I can't remember what it's called now. I'll find a link for the pig and I'll let you know for those that want to know. Right, okay, so I've made a copy of my script and I've called it get SRA robust, which is perhaps not the best name in the world, but it'll do for now. And if you remember what I had talked about, my plan was that I would use the file sizes in the SRA run table that we downloaded and check the data that we downloaded against the file sizes in that run table. And in trying to do this, I discovered 2 things. So the first thing that I discovered was that those file sizes are very slightly different to the sizes of the SRA files that you download from using Prefetch. So I used Prefetch to download the SRA file. Then I compared the size of the SRA file that I downloaded with the size in the table, and they were very slightly different to each other, which is frustrating to say the least. So any check that I could implement for the file size in the table versus the file size downloaded from the SRA would not work. But the other thing that I discovered while doing that is actually Prefetch does these checks for you. So Prefetch, when you request a particular accession, will download it to a standard location, and if you've already got a file with the appropriate name, it will check to see if that file is the right size, and if it isn't, it will download it again. So actually Prefetch does for us the thing that I was going to write a script to do. So if we look at the script that I was writing yesterday, you'll see that it was getting quite complicated, and I didn't really want to expose people too much to this script because it's a little bit of a mess. I would have tidied it up if I'd needed to, but actually I did discover that that Prefetch does this for us. So I honestly don't think that we need to do too much stuff here. So I think actually what we should do is rely on Prefetch doing it correctly, and if Prefetch completes successfully, then we can assume that it's worked. So actually, if and I'll run this and I'll show you. So this works just for at the moment this is set up just to run for the first file. So I'm getting a sequence of one number, just to let you know what that does. I'm not going to go into too much other details about this script, but this will run for SRR 114.122.15. So this one here. And if we have a look in the directory for that one, you can see that file is already downloaded, and we can see how big it is. So it's 232 meg. So we've already downloaded that file. And if I try to download it again, so actually I need to activate the right, so it's conda activate RNA-Seq. Can't spell activate. There you go. So if we activate the right environment, so if we run Prefetch and then SRR 114.122.15, it will Prefetch will check what's in my SRR 114.122.15 directory, says it's found locally, and it has 0 unresolved dependencies. Okay, so what that means is, as far as Prefetch is concerned, I've already downloaded that file. So this is effectively doing the if check that we wrote into our script yesterday for us, which is quite nice. If we look at the next one, so let's look at 1.6, because I went through and I touched all these files. So the SRA file here exists because I've created it with touch, but it's empty. So this clearly isn't the file that Prefetch would download. Okay, so if I then prefetch 1.6 rather than 1.5, what you'll find will happen this time is it checks, it says, okay, so it looks at that file and it says it's 0 bytes, so it's incomplete. The expected size is 235,407,954 bytes. It will be re-downloaded. So it realizes that the file isn't the size it should be, and it will go and re-download it for you. Hopefully what we'll get back at the end of that is a success message. So what we really want to be able to do is to have some way of checking whether Prefetch successfully completes or not. But actually, we can run this script and then we can run it a second time. And if we run it a second time, they should all just say that they've successfully completed. And hopefully then we've got all of the data. And then what we can then do is go back and remove the SRA files once we've converted them to FastQ. So I'm just going to edit this file, and I think I'm going to use Prefetch to download the SRA files, and then I'm going to use FastQ dump not to download the FastQ, but to dump out the FastQ from the SRA files that we've downloaded. And that will be more robust than the way that we've currently done it. And we'll make sure that we've got all of the data. So I think that's what I'm going to do. What I will do is I will just very quickly make some edits to this script to make it work. Again, probably offline, I think. Or maybe we'll do it now, actually. It shouldn't take too much to do it now. So let's open a new tab. Let's edit our get SRA robust script. Okay. We don't need the file sizes anymore. So let's change all of this. We don't need all of this. And then we want 4i in SRA. So there's a lot of complications there here that we really don't need. So we don't need that or that or that or that or that or that. We just need to do this. Delete that as well. And this is $I. So that will download the data. And then obviously the other thing that we want to do is we want to create the FastQ from that downloaded SRA file. So that's the other thing that we want to do in this script. But I only want to do that if the FastQ doesn't exist. Because the Prefetch will check for itself, but FastQ dump won't. So I want to put that if statement in here that I had in the previous script. So I'm going to save this and exit just so I can get the if statement from the other script. So nano get SRA data. So here's my if. So I will borrow this, copy, and then exit, and then go back to the other script and paste it in there. There we go. So if the FastQ file exists, then do nothing effectively. Otherwise, we want to convert the SRA entry, and we want to do FastQ dump. So we want to do exactly the same thing, but instead of just putting the accession here, we want to put the SRA file that we want to convert. So that's the thing that goes at the end of the FastQ dump. So that's $i.sra. So that's the SRA file that we want to convert. And that will produce the FastQ from the downloaded SRA file. So if Prefetch, assuming Prefetch runs fine, then our FastQ files will get generated. Yes. Well, assuming, yeah. And if we run this a second time, if Prefetch has worked fine, then it won't run again, and our FastQ files will already exist. So they won't get created again either. So that should be the kind of most robust way of getting this data, I think. I hope that should work relatively straightforwardly. So Prefetch downloads the SRA file from the SRA, puts it into a directory with the name of the SRA accession, and then the file inside the SRA accession, inside that SRA accession directory is the SRA accession again, .sra, and then we're using FastQ dump to convert that SRA entry from the SRA format into FastQ format. And that should be sufficient and should be more robust than FastQ dump for the accession itself. Now, I feel, and some of you may feel as well, slightly that we're tying ourselves in knots here. And we slightly are, I think. I'm sure there are more efficient ways of doing this. But to me, I would like to make sure that everybody has the same data. And my worry with doing it just by FastQ dump is that it clearly doesn't work successfully all of the time, and it works unsuccessfully enough, often enough, that I don't trust that we all end up with the same data. Whereas Prefetch, I think, is more robust. And so we will, in theory, all end up with the same data. What I haven't been able to find, and some of you, there's some chatter in the live chat I can see, I haven't been able to find a direct link to the FastQ files. And if there is a direct link to the FastQ files, then getting those with wget is probably actually the simplest way of doing this, and is going to be less error prone. So if we could track down a direct link to the FastQ, then that may well be the most sensible way of doing it. But for now, this is the best that I can think of. Again, what I think I will do is I'll check that this script works. I'll put it up online so that when we start actually processing this data in Anger, we can all be sure that we're working from the same data, so that we should then all get the same results. Because my great fear is that everybody ends up with slightly different results, because actually everybody's working from slightly different data. Okay, so we'll put that aside for the minute. I've been wrapping myself around trying to get this done correctly for a few days now, and I feel like we're too far down the rabbit hole and we're using more time than we really need to do it. And I can see my live stream numbers dropping off as I continue to talk, so I think we'll move on to talk about FastQC, which is what I said we were going to talk about today. So that's what we'll do now. So if you remember yesterday, we ran FastQC. I left the session at the end of the session. Excuse me. There we go. I was running FastQC over my 48 FastQ files that I had managed to download, whether correct or incorrect, and we'll come back to that another time. But so I left FastQC running over those files, and there you go. Hey presto! Actually, let's just check. We should have 48 HTML files. That's those. So count the number of lines. 48. So 48 HTML reports, which is the output of FastQC. So that's our reports, and we can amalgamate those into one single report using MultiQC. We talked about this yesterday as well. So if we run MultiQC with the input directory of our FastQC output directory, it may... Why is it doing... Oh, because I'm not in the conda activate. Because I've opened another terminal. There we go. Run again. MultiQC, FastQC output. Then it will find those 48 reports, and it will produce us an amalgamated report of all of those 48 reports. So it's done that because the file multiqc report.html already existed. It's created multiqc report 1.html for us. So we'll take a look at that later. But what I wanted to do first of all was take a slightly more detailed look at a single FastQC report, and try to talk a little bit about some of the assumptions that FastQC makes when it's creating the assessment of the FastQC report for you. So what I've got here is again the FastQC report that we generated yesterday. So this is for that 2215 file for the first FastQ file in our set of 48, and I'm just going to run through this one report as an example of the output of FastQC. So FastQC produces you this nice HTML file. On the left hand side you've got a navigation pane, so a sort of general summary with all of the reports that FastQC has generated, and these should hotlink to the places in the file, but they don't appear to be working, which is slightly irritating. But anyway, so that's each one of these is one of the reports that's been generated by FastQC, and on the left hand side of that you have a little icon which tells you whether FastQC considers that this particular test has passed or failed, or whether FastQC gives you a warning for that particular test. And a lot of people's first instincts when they look at these reports is that they want to eliminate those red crosses. They see the red crosses and even the orange exclamation marks, and they want to get rid of those things. They think that those things must be indicative of some sort of bad quality, and so they want to get rid of them. And what I want to talk a little bit about is the assumptions that FastQC makes in producing those green ticks and red crosses, and whether those assumptions are always appropriate or not for the data that we've got in hand. So let's begin at the top and we'll work our way down. Okay, so the first thing is just a table of some basic statistics telling you what the file is that you've processed, the encoding of the quality information, which we can maybe talk a little bit about in a second, how many sequences there are in the file, so just over 4 million or 4.25 million in this case. There's no sequences flagged as poor quality. They have length ranging between 35 and 151 bases, and they've got 49% GC content. So that's a useful set of kind of core statistics about this particular file. You will always get a green tick next to this one because it's not really testing anything, it's just giving you information. Okay, so then the first report of any real substance, and let me just try and make it a little bit smaller so it all fits on the screen. There we go. Is this per base sequence quality, which is a box plot, which is summarizing the sequence quality at a particular position in the read across your whole experiment. So here we've got 4.25 million sequences. So this box here is summarizing the quality of all of the bases at the first position in all 4 million of those reads, and it's showing you the distribution of quality in those reads, where what it means by quality is the Phred scaled Q score that is present in the FastQ file. So let's have a quick look at the FastQ file, and I can show you what I mean by that. So here we go. If we take a quick look at this FastQ file, so let's use zless just to look at the first page of the FastQ file that we've got this report for. So this is FastQ file. The entries are spread across 4 lines in FastQ. So the first line is the sequence identifier. So this is from the SRA. So the identifier is an SRA identifier. So the SRA accession, run accession plus a .1, .2, .3 for the number of read that you're in the file. So that's an SRA identifier. The SRA FastQ also tells you the length of the read in question. If this was the Illumina data, so if this was directly from an Illumina sequencer, this identifier line would look quite different, and it would contain a lot of metadata about the sequencing. We can talk more about that at some point if it's appropriate, but it doesn't matter right now because we don't have that information available to us. The second line is then the sequence data itself. So these are your A's, T's, G's, C's, and N's, which make up your called bases from this sequencing read. So this is your data effectively on this line. The third line is then a separator between the sequence information on line 2 and the quality information that's encoded on line 4. Okay, so this is just a separator. It's usually just a plus character, and it always begins with a plus character. You can have other stuff on there, but the way that we've encoded the FastQ is we've got no other information on that line, so it's just a plus for a separator. And then the fourth line of the entry, the fourth and final line, is the quality information. And so this is ASCII encoded. It's actually numeric data, but it's encoded as ASCII text. So you get 1 character per base of quality information, and it is possible to translate that letter into a number. And different FastQ formats from different eras will be encoded in slightly different ways. So what we're seeing here, from the look of it, is we know that this is an XSeq run. So we know that from the metadata in the SRA and at Geo that this was an XSeq run, and you can actually tell at a glance as well that this must be a recent Illumina sequencing run, because the quality, even from this one page of data, the quality is only taking certain values. So I can only see for maybe five different values for quality on this one page. So I can see A's, and I can see E's, and I can see hashes. So that's 3. But I can also see less than characters and /. So I can see 5 different characters on here. And that's because on modern Illumina sequencers, the quality scores are binned into so they can only take particular values. So rather than having the range of all values from 1 through to 41, which is the usual range, on modern Illumina base callers, your quality can only take certain values. It's limited to a certain number of values. I think with the NextSeq, it's something like 7 different values that the quality scores are binned into. And that is mainly because it means that this file will compress more efficiently. So your resulting gzipped FastQ files will be smaller with binned quality than with unbinned quality. So that's why the quality score is only taking particular values, but each of these ASCII values, so the A and the hash and the E and the other values that you find, can be interpreted as being a numerical value by using a lookup table. And actually, the best place to find those lookup tables is on the Wikipedia entry for FastQ. So if you Google FastQ and jump to the Wikipedia entry, not the Illumina page, the Wikipedia page about FastQ, down here you will find, if we scroll down a bit, you will find a lookup table for those quality values. And by finding out which scale we're working off, and if we look at our FastQC report, we can see that we're working off Illumina 1.9 format encoding. And we go back to the Wikipedia page. So that's Illumina 1.8 plus, or alternatively Sanger as well. They're kind of the same scale. But we're looking at this set of L's here. So you can read down from, by looking at the L's, you can say, well, and these numbers at the bottom, which are the same colour. You can say, okay, well, the hash here, that is the equivalent to a 2, a quality of 2 on this quality scale, which is a low quality base call. And the other things that we see in that file, so we see hashes, we see A's, and we see E's. So back to the lookup table, the E, for example, here. Okay, so we skim down to the bottom here, and that's a quality of mid-30s. So about 36, 35, 36, which is a high quality base. Okay, so the Fred scale of quality is it's a minus 10 times log to the base 10 of the probability of error. So the higher the Fred scaled quality score, the lower the probability of error that you have for that particular base call. And the way that you translate it is a Fred score of 30 is a probability of error of 1 in 1,000. A Fred score of 20 is tenfold lower, so probability of 1 in 100. And a Fred score of 10 is 1 in 10. It's another tenfold lower. So what you're looking for is as high a Fred scaled quality score as possible, because the higher the Fred scale quality, the lower the probability of error. And so to go back to our FastQC report, here we are. So what this is telling us about the quality here is that, generally speaking, the quality of our sequencing is very high. And you can also see in this box plot, you can see the effect of the binning, because bases can quality scores can only take certain values. These box plots are very discrete. So you see that basically the mean for the first 5 bases is 32, which is an A. and there's no box around that 32 because there's really almost every base at that first position will have a quality score of an A, and we can just have a look at our reads, and you can see just on this first screen, the first base here, or in fact the first 5 bases, they all have a quality of A. So that is bearing out what we're seeing in that box plot, those A's that we see in the FastQ, because A is 32. So we're seeing very little variance around that 32. So we can those discrete bins of quality are represented in this box plot, and then as we scan across the sequencing, you see that it starts at 32 and then jumps up to 36 after about 5 bases. That's very typical, and it's to do with calibrating the sequencer, and cluster finding in those early bases reduces the quality somewhat, but not too worrisome here. And then after you've done your first 5 or 6 cycles, then the quality jumps up, and then gradually over the length of the read, the quality will drift slightly downwards. And that's to do with, as your reads get longer, your clusters get out of phase, and your surety of your base calls gets lower, so your quality will drop off slowly, and that is a feature of Illumina sequencing, and you always see it. What FastQC is looking for with this report, in order to get a green tick, is that the median, that is the red line in the middle of the boxes, never falls out of the green zone. So as long as it's in this green, as long as the red line is in this green zone, then FastQC is happy. If the median falls out down below that green zone, then you might have reason to take a slightly closer look at what's going on with the quality of your sequencing. Bearing in mind that the green line here is drawn at 28, so a quality of 30 is a 1 in 1,000 probability of error for that particular base call. So I think we can say that the quality of this data is pretty high. Okay, so moving on. So that's the box plot, and a general discussion about Q values in FastQ files. Just to move on from that then to the next one, per sequence quality scores. So this is, instead of taking per base like this, where you're looking at all of the reads at position 1 in all of the reads and looking at summarizing the quality at that position and then doing the same at position 2 and so on. For this, you're taking the average quality for the whole read, and then you're drawing what is essentially a density plot for the average quality across all of your reads in your whole experiment. And what you expect to see for this is that on average, your read should have a relatively high quality. And that is indeed what we're seeing, that on average, almost all of our reads, and certainly more than half of our reads, have an average quality above 30. And that tails off very quickly, that there's very few reads that have an average quality below, say, 20, if any, in fact, if any reads at all. So this is showing you that, okay, per base, we can be quite confident in the quality, and then we're not seeing a hugely dramatic drop-off in quality at the higher cycle numbers. But we can also say that we don't have a huge number of really poor quality sequences overall. So you do find in some sequencing runs that most of your sequences are fine, but that you have a bunch of sequences where effectively the base calling has failed, and you've got a run of ends, and those ends have very low quality, and they will show up as a spike down at the lower end of a bunch of reads of poor quality. And that's what you don't want to see in this plot. You don't want to see a spike down at the lower end, indicating that some of your experiment has failed, that the sequencing run has just not quite worked out the way that you'd hoped. Okay, so on to our first cross, and that is the per base sequence content. So this is again looking at positions in reads, so looking at position 1 in every single read in the experiment, and figuring out the balance of the different bases at that position. So the balance of A's, T's, C's, and G's at that position. And what you find is let's start with what FastQC expects. What FastQC expects to find for this particular report is parallel lines, so that the quantity of A's, T's, C's, and G's doesn't vary across the read. It doesn't matter what value those take. So it might be 25% of each, or it might be it might vary from that, 30% of one and 20% of another. But it would expect FastQC expects parallel lines as an ATCG distribution across the length of the reads. What you will find if you analyse a lot of sequencing data, and I have analysed a lot of sequencing data over the years, is that actually this plot is very rarely parallel. And there are lots of reasons for that. So the assumption of parallel lines comes from if you were doing an unbiased genome sequencing experiment. So you would expect that your fragments are randomly generated from your genome, that there's no bias in the way that they're distributed, and so the distribution of base content in your reads should be the same as the distribution of base content in your genome. But that's not what we've done here in this experiment. We've not sequenced the genome, we've sequenced the transcriptome, but also the way that our fragments have been generated is also not entirely random. So when you produce your cDNA, your first strand cDNA from your fragments of messenger RNA, in order to prime that reverse transcription reaction, the standard library prep methods uses random hexamers to try and prevent introducing any bias in mRNAs that are more or less likely to get converted into cDNA. Turns out that process of random priming is not entirely random, and that's what's being exhibited in the first dozen or so bases in this data. So we're not seeing parallel lines because we're seeing the pattern of bias in that random hexamer priming. That I entirely expect to see that pattern in an RNA-Seq experiment. The pattern that we're seeing in this file comes as no surprise to me whatsoever. It's what I expect to see. People's first instinct when they look at their first FastQC report from an RNA-Seq experiment is something weird has happened in the first 10 bases. I must trim those 10 bases off so that I get rid of that weirdness. Actually, you really don't need to. Although there's a pattern of bias here, what you will find is as we go through these reports, when we look at the MultiQC, for instance, that this pattern is the same across our whole experiment. It's not different between our different samples, and it's not even with you might expect that with the 4 samples, the 4 SRA accessions taken from a single sample, but it will also not be different between all of our 48 files. This pattern of bias will repeat again and again and again. And because of that, these bases, they may be biased, but they come from our transcriptome, and they will aid the mappability of these reads when we're mapping to the transcriptome next week. So we don't need to trim this data. There's no reason to try and get rid of this particular pattern. And this is an example of where FastQC goes into this analysis with certain assumptions set, and those assumptions aren't necessarily going to be suitable for all types of experiment. So you'll see similar patterns of bias in modern bacterial genome sequencing experiments where the genome has been fragmented using tagmentation, so using an enzymatic process. You'll see a pattern of bias at the beginning of those reads as well. The same thing applies, that those reads come from your genome, that's genomic DNA that you've sequenced. The enzymatic fragmentation has introduced a bias, but that's not a bias that we necessarily need to worry too much about. So this particular report in FastQC will very often fail. It's very rarely something you actually need to worry about. I do find that trimming in most high throughput sequencing experiments, trimming really isn't required. There are very few cases where I would really favour trimming. The major one is trimming adapters. So if you have a significant adapter content, and we'll come to that later, if you have significant adapter content, that is worth trimming. If it's anything else, like quality-based or these sorts of patterns of bias, it's almost never worth trimming that data. You do more harm to your experiment than good. Okay, moving on. Per sequence GC content, FastQC expects to see that all of the data is drawn from a single GC distribution. That again is predicated on unbiased genome sequencing. That's not what we're doing here. So you don't necessarily expect to see that because we've got reads that are generated from mRNA produced from genes across the genome, and those genes may have subtly differing GC content. So you're not necessarily drawing all of your reads from a single GC content distribution graph. So you wouldn't necessarily expect the observed distribution in your reads to match the theoretical distribution if we had one single value of GC content for our entire experiment. So again, although we've got a warning because we see slight deviations from the perfect blue curve, it's not significant enough to worry about, and it's not something to be concerned with anyway. Okay, per base N content, if you had particular cycles in your sequencer that failed, you may see spikes at particular positions, which would be cause for concern, and we don't see that here. So this is fine, and we can proceed with this. Sequence length distribution, again, this is predicated on the fact this is Illumina sequencing, and the assumption always was that an Illumina sequencer produced fixed length reads. So all of your reads would be 150 bases. There would be none that were shorter, none that were longer, and you would see this. The perfect distribution here would be that every single read was the same length. With modern Illumina platforms, the trimming is done by the base caller. It's done effectively on the device. And so you do see variable length reads from modern Illumina experiments. And that's what we're seeing here. A lot of our reads are 150 ish bases long, which is the expected length, the number of cycles the sequencer has been run for, but there's a goodly proportion that are shorter, and that's because they've been quality trimmed on the device. So the base caller, BCL to FastQ, which is the program which produces the FastQ files, usually on BaseSpace, Illumina's cloud platform, has done trimming at the point at which those FastQ files have been produced. Again, that is to save space. That's because the throughput of Illumina machines is such that if you saved all of that data, you would have more than you really know what to do with. So again, we have a warning here, but we don't need to worry too much about that. This is expected. Sequence duplication is, again, the assumption here is, again, for unbiased genome sequencing, where you expect a low level of duplicated reads, and duplicated reads is indicative of PCR bias. With RNA sequencing, you're sequencing a hugely enriched pool of stuff. So because you're sequencing mRNA, which is, for a start, is only 2% of the genome, but also there are certain signals, there are certain messenger RNAs in that pool of data that are hugely enriched. So there are certain genes that are expressed at very high levels that will produce a reasonable amount of the data that we're seeing. So you expect to see duplication in RNA-Seq experiments. You expect that that a lot of your reads are going to be duplicate reads. And you even expect to see quite a lot of duplication. The level here is relatively high. So there's only about 30% of the reads overall are unique, which is fairly high level of duplication, which is indicating that maybe the input amount of RNA into this experiment was a little bit low. So maybe we've got a bit more PCR duplication than we would really like. but as long as it's relatively consistent across the whole experiment, it doesn't matter too much. So I think we can be relatively hopeful that this experiment is okay. And although, again, FastQC doesn't like this, it's because the assumptions it's making are predicated on something other than than it being an RNA sequencing experiment. Okay, for overrepresented sequences, again, you can sort of relate this to the duplicated sequences above. If you've got a high level of duplication, then you're likely to have a high level of overrepresented sequences. As long as none of these are hitting adapters, then it's probably okay in an RNA-Seq experiment. So again, we're seeing certain things here. It may be that we might want to investigate these more closely, but for now, I think this is fine, and none of them are adapters, which is the most important thing. None of them are strings of ends either. That would be another cause for concern. And then finally, adapt content. So if we were seeing the Illumina universal adapters popping up as the read length progressed, it would show us that our fragments were shorter than our sequencing reads, and that would be a reason. That would be a strong reason to trim these reads, to do adapter trimming of these reads, and that's the one kind of trimming that I really would advocate in most cases, in most high throughput sequencing experiments. Yes, good point in the comments. If this is a sample infected with a virus, then they may well be reads coming from the virus, which is obviously a much smaller genome and will be very prevalent in a virally infected sample. So that is a very good point. It may well be that we've got viral reads in here. I don't know if this is a virally infected sample or not, actually, but yes, that's a very good point. Could easily be the case. But again, your knowledge of the experiment is trumping FastQC's expectations, FastQC's assumptions. And that's always the thing that I would say when looking at these reports is your knowledge of the experiment that you have done, especially if it's your experiment, is always going to trump FastQC's expectations of your data. So go into assessing these reports in a kind of knowledge-led way. Don't just look for the red crosses and assume that you want to eliminate them. What I would say for this experiment, despite the fact that we've got two crosses and a few warnings, I don't think there's any significant action that's required for this sample. I think this is a perfectly fine RNA-Seq sample that isn't showing me any cause for concern. Okay, so then the final thing that I want to do today is take a very quick look at the MultiQC report. So the MultiQC is effectively replicating what FastQC is doing, but it's showing you the reports for all of your files in one go. So if I grab my Finder window up that I had yesterday, so here's Finder, and here's my new MultiQC report that we produced. At the beginning, or about half an hour ago, I guess now. So I dragged that into my browser, and we'll take a look at that. So here's the MultiQC report. So yesterday we had just 2 reports in our MultiQC report. Now we have all 48. Okay, so we can scan down this and see where we have any causes for concern. So you can see a higher level of duplication in this batch of 4 here, but also about half again as many reads. So you would expect higher duplication when you've sequenced to greater depth. So that's not unreasonable. Here's another sample where again the duplication jumps up, but again the coverage has jumped up. So once again, not an unexpected finding. So I think those first ones are one batch, and then here's the other batch. I think potentially this is where the status of viral infection changes, is this second batch, where you see that the duplication rate jumps down a bit for this second batch. and again kind of keeps tabs with the coverage. So the more reads you've got, the higher the duplication level, but there's a fairly direct relationship between those two things. So again, no real cause for concern there. And that's the equivalent information, but in little charts, and you can show that out as percentages, and that's of certain value, I suppose, in terms of information. Quality is pretty much the same across the board, very little deviation, and they're certainly all fine. Same for per sequence quality. I told you that those patterns would be very similar across the whole experiment, and certainly, I mean, maybe we're seeing that these two sets of samples are a little bit batchy. That the first set is slightly different from the second set, but there's, again, these are RNA-Seq samples. These are the patterns we expect to see. There's no huge cause for concern here. Okay, so real change between, and again, I suspect this is between batch 1 and batch 2 of these experiments. So I'm pretty sure that if we go back up here, this first half of the samples is one condition and the second half is the other condition. So I think we're seeing the effect of the different conditions in the QC here. So yes, again, nothing too dramatic going on here. Per base N, not too bad. Never more than 1%. Sequence length, again, the same as before. Duplication rates, well, we knew what that was going to be from the table above. And then overrepresented sequences. So, okay, I would say if we are looking at viral reads at all, we're looking at them in the second set, because we've got a lot more overrepresented sequences in the second set than we have in the first. And I would put good money on that being the virus, to be honest. So I think that's where we're seeing that being exhibited. And then adapter content is fine. There's a little bit, but again, it never really gets above 1%. So let's not worry too much about that. So I think overall, the quality of this experiment is very good. It is clearly an experiment of two halves, and that's fine. We've got two conditions. That's okay. And so I think that that we can work with this data. It looks okay to me, so that's good. People are asking about the viral genome. The SARS-CoV-2 genome is on NCBI. There's a RefSeq for the Wuhan strain. I've got that accession somewhere. I'm just trying to find where, because I've been doing a little bit of analysis against the genome for somebody. So let me just find it. There we are. That's the... Right, bear with me a second. There we go. This is the NCBI. This is the GenBank entry for the SARS-CoV-2 genome. So it's about 30,000 bases. And it's NC_045512. So that's the viral genome, should anybody be interested. Okay, which I'm sure we will come back to. We're going to map to the human transcriptome, but it might make sense to map out to the viral genome as well at the same time. Right, okay, so that is FastQC in excruciating detail. I hope that has been informative and useful. And I know we've not done very much practical today, but today was going to be about kind of going through those results from previous from our previous lesson. The Prefetch version of the script I will polish up and I will post on GitHub, and I'll post a link to it in the description of this video when I when I archive this video. So if you want to run the prefetch version of the script, then I suggest that might be a thing that's worth doing. And I will be doing that over the weekend. So I will be starting with on Monday with the data which I have acquired using Prefetch to download the SRA files, and then FastQ dump to convert those SRA files that I've downloaded to FastQ files. So that's what I'm going to do. I will do that over the weekend. Okay, so everybody have a lovely weekend. Have a break from all of this, and we'll come back refreshed on Monday. I will have done a day's work on Monday, so it may be hopefully we'll be good to go, and we'll start to think about the processing that's required. Now we've looked at the QC, and I think our conclusion from today can be that the QC is pretty good. Then we're ready to proceed. We're ready to do some stuff with this data, do some bioinformatics in anger with this data. So we will look at doing that next week. And so I will let the pig play us out, and I will see you all on Monday. So have a lovely weekend. Bye, everybody. Bye.